I'm Becca.
Mi nombre es Pablo.
My name's Sam.
Thanks for watching us on DPS TV.
So we think of DNA as the instructions for a living organism, the blueprint for a living organism. And that's because DNA codes for the proteins that allow that organism to be built. But how it codes for those proteins is something of interest. So in this video, we're going to talk about the nucleotide base pairings, essentially the pairings that happen within DNA that help to build the code, that code for all the proteins in any given living organism. So if we could jump to my screen. So this is just a simplified diagram of DNA. We know that DNA is a double helix, so it's two molecule strands wrapped around each other. And it kind of looks like a ladder, and the rungs of the ladder are made of what are called nucleotide bases, and each rung is actually two bases paired together. So the four bases that make up the rungs of the ladder in DNA are adenine, thymine, cytosine, and guanine, and we abbreviate those A, T, C, and G. And it's these four bases contained within DNA that allows DNA to contain a code, and it's that code that then later is transcribed and then translated into proteins, and that's something that we covered in another video. But DNA itself stores this genetic code as two strands, and the strands have to be locked together. And the way that DNA is able to be locked together is because certain bases pair with certain other bases. So adenine always pairs with thymine. So we'll say that A pairs with T, and we often just show it like this. We show sort of an A-T to show that they pair together. And in DNA, cytosine pairs with guanine. So if we imagine that we have a strand of DNA, and let's say we know one side of that strand of DNA, we could write it as A, T, G, C, T, T, A, G, G, C, A, T. So this is just sort of an imaginary section of DNA, a strand of DNA, and again, it's just one side of this two-sided molecule. And so each of these letters, again, represents a nucleotide base, one of the chemicals that makes up the code that codes for these proteins. So again, this is one strand of DNA. We can imagine that back behind these bases is the backbone here. It's a sugar phosphate backbone. And the backbone just provides structure. It doesn't actually do anything in terms of helping with the code. It just holds the DNA molecule together. But imagine that the other half of the DNA molecule has to match, the code on the other strand of the DNA molecule has to match with this strand of DNA. So if we know one strand of DNA, we can predict what the other strand will be. So in this case, since we have an A, we know that that A will pair with T, so the other strand will start with a T. Then we know that the T will pair with an A, the G will pair with the C will pair with a G, and so on. And so it's easy, fairly easy, I should say, to predict a second strand of DNA, or a pairing strand of DNA, a complementary strand of DNA, given the first strand. So I'll just go a little bit further here. DNA has a cousin, another nucleic acid, called RNA. RNA is very similar to DNA, but there's a few distinctions. RNA is a little bit shorter than DNA, or I should say significantly shorter. RNA is just a single strand. It's not a double strand or a double molecule. And RNA has the ability to leave the atomic nucleus, or the cell nucleus, and interact with amino acids to form proteins. So RNA has a code just like DNA, made of 4 nucleotide bases. And RNA contains 3 of the same nucleotide bases as DNA. But RNA actually replaces thymine, the T base, with another base, uracil. So if we imagine RNA, and let's do this, let's change color here. So I'll do RNA in red down here. If we had an RNA molecule, RNA still has adenine. Let's see, RNA. Here, I'll just put DNA. So if I had an RNA molecule, that RNA molecule has adenine. But in RNA, adenine will pair, instead of pairing with thymine, it'll pair with a chemical called uracil, a different nucleotide base, and cytosine will still pair with guanine. So something that can happen, or something that happens frequently inside of cells, is that DNA is transcribed into RNA. So a section of DNA will become a segment of RNA, and that segment of RNA will then leave the cellular nucleus to code for a protein. And so what we can do is we can imagine, and I'll do this in black and red so we can see the distinction here. But if we have a strand of DNA, and I'll again just come up with sort of a made-up strand. If we have a strand of DNA that goes thymine, guanine, cytosine, 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 thymine, adenine, guanine, cytosine. That's a strand of DNA, and we can tell that it's a strand of DNA because it contains the base thymine, that T. If this strand of RNA, if this strand were transcribed into a section if this strand of DNA, excuse me, was transcribed into a section of RNA, then we would get this section of RNA transcribed. So it would be, pairing with the T would be an A, pairing with the G would be a C, the C pairs with guanine, guanine, guanine. So it's really the same system as building a complementary strand of DNA. But when you come to an A, that becomes a U. A pairs with uracil in RNA, and guanine pairs with cytosine, and cytosine pairs with guanine. So again, it's this distinction right here that when RNA is formed, any would-be thymine base instead becomes a uracil. But in either case, whether we're talking about DNA or RNA, there are 4 nucleotide bases that make up a 4-letter code, and it's that 4-letter code repeated in infinite variations over these giant DNA molecules that code for all the proteins in any given living organism. So hopefully that helps to clear up a little bit how nucleotide bases pair in nucleic acids.